Introduction to biochemistry. Importance of carbon. Importance of water. Carbohydrates. Lipids. Proteins. Nucleic acids. Conjugated molecules. Introduction to biochemistry. Biochemistry is a branch of biology which deals with the study of chemical components and the chemical processes in living organisms. A basic knowledge of biochemistry is essential for understanding anatomy and physiology. For example, photosynthesis, respiration, digestion, muscular contraction can all be described in biochemical terms. All living things are made of certain chemical compounds, which are generally classified as organic and inorganic. Most important organic compounds in living organisms are carbohydrates, proteins, lipids, and nucleic acids. Among inorganic substances are water, carbon dioxide, acids, bases, and salts. Typically, an animal and bacterial cell consists of the following chemicals. The survival of an organism depends upon its ability to take some chemicals from its environment and use them to make chemicals of its living matter. For this reason, cells of every organism are constantly taking in new substances and changing them chemically in various ways, like building new cellular materials and obtaining energy for their needs. Life of an organism depends upon ceaseless chemical activities in its cells. This chemical activity is maintained with a high degree of organization. All chemical reactions taking place within a cell are collectively called metabolism. Metabolic processes are characterized as anabolism and catabolism. Those reactions in which simpler substances are combined to form complex substances are called anabolic reactions. Anabolic reactions need energy. Energy is released by the breakdown of complex molecules into simpler ones. Such reactions are called catabolic reactions. Anabolic and catabolic reactions go hand in hand in living cells. Complex molecules are broken down and the resulting smaller molecules are reused to form new complex molecules. Interconversions of carbohydrates, proteins, and lipids that occur continuously in the cells are examples of coordinated catabolic and anabolic activities. Importance of carbon. Carbon is the basic element of organic compounds. Due to its unique properties, carbon occupies the central position in the skeleton of life. Carbon is a tetravalent. It can react with many other known elements forming covalent bonds. When a carbon atom combines with 4 atoms or radicals, the 4 bonds are arranged symmetrically in tetrahedron and result to give a stable configuration. The stability associated with the tetravalency of the carbon atoms makes it favorable element for the synthesis of complicated cellular structures. Carbon atoms can also combine mutually, forming stable, branched, or unbranched chains or rings. This stability of carbon is responsible for the vast variety of organic compounds. Carbon-to-carbon bonds form a skeleton of organic compounds. Carbon combines commonly with hydrogen, oxygen, nitrogen, phosphorus, and sulfur. Combination with these and other elements contribute to the large variety of organic compounds. Carbon and hydrogen bond is the potential source of chemical energy for cellular activities. Carbon to oxygen association in glycosidic linkages provides stability to the complex carbohydrate molecules. Carbon combines with nitrogen in amino acid linkages to form peptide bonds and form proteins, which are very important due to their diversity in structure and function. Large organic molecules or macromolecules such as cellulose, fats, proteins are generally insoluble in water. Hence, they form structures of cells. They also serve as storage for smaller molecules like glucose, which in turn are responsible for providing energy to the body. Smaller molecules such as glucose, amino acids, fatty acids, and many others serve either as a source of energy or as subunits to build macromolecules. Some small molecules are so unstable that they are immediately broken down to release energy. For example, ATP. Such substances serve as immediate source of energy for cellular metabolism. Importance of water. Water is a medium of life. It varies from 65 to 89% in different organisms. Human tissues contain about 20% water in bone cells and 85% in brain cells. Almost all reactions of cell occur in the presence of water. It also takes part in many biochemical reactions, such as hydrolysis of macromolecules. It's also used as a raw material in photosynthesis. Solvent properties. Due to its polarity, water is an excellent solvent for polar substances. Ionic substances, when dissolved in water, dissociate into positive and negative ions. Non-ionic substances having charged groups in their molecules are dispersed in water. When in solution, ions and molecules move randomly and are in a more favorable state to react with other molecules and ions. It is because of this property of water that almost all reactions in cells occur in aqueous media. In cells, all chemical reactions are catalyzed by enzymes, which work in aqueous environment. Non-polar organic molecules such as fats are insoluble in water and help to maintain membranes, which make compartments in the cell. Heat capacity. Water has a great ability of absorbing heat with minimum of change in its own temperature. The specific heat capacity of water, the number of calories required to raise the temperature of 1 gram of water from 15 to 16 degrees is 1. This is because much of the energy is used to break hydrogen bonds. Water thus works as a temperature stabilizer for organisms in the environment and hence protects living material against sudden thermal changes. Heat of vaporization. Water absorbs much heat as it changes from liquid to gas. Heat of vaporization is expressed as calories absorbed per gram vaporized. The specific heat of vaporization of water is 574 kilocalories per kg, which plays an important role in regulation of heat produced by oxidation. It also provides cooling effect to the plants when water is transpired. Or to animals when water is perspired. Evaporation of only 2 ml out of 1 liter of water lowers the temperature of remaining 998 by 1 degree C. Ionization of water. The water molecules ionize to form hydrogen and hydroxyl ions. This reaction is reversible, but an equilibrium is maintained. At 25 degrees C, the concentration of each of hydrogen and hydroxyl ions in pure water is about 10 to the power minus 7 moles per liter. The hydrogen and hydroxyl ions affect and take part in many of the reactions that occur in the cells. Protection. Water is an effective lubricant that provides protection against damage resulting from friction. For example, tears protect the surface of eyes from rubbing of eyelids. Water also forms a fluid cushion around organs that helps to protect them from trauma. Carbohydrates. Carbohydrates occur abundantly in living organisms. They are found in all organisms and almost all parts of the cell. Cellulose of wood, cotton, and paper. Starch is present in cereals, root tubers, cane sugar, and milk sugar are all examples of carbohydrates. Carbohydrates play both structural and functional roles. Simple carbohydrates are the main source of energy in the cells. Some carbohydrates are the main constituents of cell walls in the plants and microorganisms. The word carbohydrate literally means hydrated carbons. They are composed of carbon, hydrogen, and oxygen. And the ratio of hydrogen and oxygen is the same as water. Their general formula is CX H2OY, where X is the whole number from 3 to many thousands, whereas Y may be the same or different whole number. Chemically, carbohydrates are defined as polyhydroxyaldehydes or ketones. Or complex substances which on hydrolysis yield polyhydroxyaldehyde or ketone subunits. Hydrolysis involves the breakdown of large molecules into smaller ones, utilizing water molecules. The sources of carbohydrates are green plants. These are primary products of photosynthesis. Other compounds of plants are produced from carbohydrates by various chemical changes. Carbohydrates in cell combine with proteins and lipids, and the resultant compounds are called glycoproteins and glycolipids. Glycoproteins and glycolipids have structural role in extracellular matrix of animals and bacterial cell wall. Both these conjugated molecules are components of biological membranes. Classifications of carbohydrates. Carbohydrates are also called saccharides, derived from Greek word saccharon means sugar, and are classified into three groups: monosaccharides, oligosaccharides, polysaccharides. Monosaccharides. These are simple sugars. They are sweet in taste and easily soluble in water, and cannot be hydrolyzed into simpler sugars. Chemically, they are either polyhydroxyaldehydes or ketone. All carbon atoms in monosaccharides except one have hydroxyl group. The remaining carbon atom is either a part of aldehyde group or a keto group. The sugar with aldehyde group is called aldo sugar, and with the keto group is keto sugar. In nature, monosaccharides with 3 to 7 carbon atoms are found. They are called trioses, tetroses, pentoses, hexoses, and heptoses. They have general formula CH2O, where N is the whole number from 3 to 7 thousands. Two trioses mentioned above are intermediates in respiration and photosynthesis. Tetroses are rare in nature and occur in some bacteria. Pentoses and hexoses are most common. From the biological point of view, the most important hexose is glucose. It's an aldose sugar. Most of the monosaccharides form a ring structure when in solution. For example, ribose will form a 5-cornered ring known as ribofuranose, whereas glucose will form 6-cornered ring known as glucopyranose. In free state, glucose is present in all fruits, being abundant in grapes, figs, and dates. Our blood normally contains 0.08% glucose. In combined form, it's found in many disaccharides and polysaccharides. Starch, cellulose, glycogen yield glucose on complete hydrolysis. Glucose is naturally produced in green plants, which take carbon dioxide from air and water from soil to synthesize glucose. Energy is consumed in photosynthesis, which is provided by sunlight. As indicated in the equation, energy is consumed in the process, which is provided by sunlight. This is why the process is called photosynthesis. It's noteworthy that for the synthesis of 10 grams of glucose, 717.6 kilocalories of solar energy is used. This energy is stored in the glucose molecules as chemical energy and becomes available in all organisms when it is oxidized in the body. Oligosaccharides. These are comparatively less sweet in taste and less soluble in water. On hydrolysis, oligosaccharides yield from 2 to 10 monosaccharides. The ones yielding 2 monosaccharides are called disaccharides. Those yielding 3 are called trisaccharides, and so on. The covalent bond between two monosaccharides is called glycosidic bond. Physiologically important disaccharides are maltose, sucrose, and lactose. Most familiar disaccharides is sucrose, which on hydrolysis yields glucose and fructose, both of which are reducing sugars. Its molecular formula is C12H22O11. Polysaccharides. Polysaccharides are the most complex and the most abundant carbohydrates in nature. They are usually branched and tasteless. They are formed by several monosaccharide units linked by glycosidic bonds. Polysaccharides have high molecular weights and are only sparingly soluble in water. Some biologically important polysaccharides are starch, glycogen, cellulose, dextrins, agar, pectin, and chitin. Starch. It's found in fruits, grains, seeds, and tubers. It is the main source of carbohydrates for animals. On hydrolysis, it yields glucose molecules. Starches are of two types, amylose and amylopectin. Amylose starches have unbranched chains of glucose and are soluble in hot water. Amylopectin starches have branched chains and are insoluble in hot or cold water. Starches give blue color with iodine. Glycogen. It's also called animal starch. It is the chief form of carbohydrates stored in animal body. It is found abundantly in liver and muscles, though found in all living cells. It's insoluble in water and gives red color with iodine. It also yields glucose on hydrolysis. Cellulose. It is the most abundant carbohydrate in nature. Cotton is the pure form of cellulose. It's the main constituent of cell walls of plants and is highly insoluble in water. On hydrolysis, it also yields glucose molecules. It's not digested in human digestive tract. In the herbivores, it is digested because of microorganisms, bacteria, yeast, and protozoa in their digestive tract. These microorganisms secrete an enzyme called cellulase for its digestion. Cellulose gives no color with iodine. Lipids. The lipids are a heterogeneous group of compounds related to fatty acids. They are insoluble in water but soluble in organic solvents such as ether, alcohol, chloroform, and benzene. Lipids include fats, oils, waxes, cholesterol, and related compounds. Lipids as hydrophobic compounds are components of cellular membranes. They are used to store energy because of the higher proportion of carbon-hydrogen bonds and a very low proportion of oxygen. Lipids store double the amount of energy as compared to the same amount of any carbohydrate. Some lipids provide insulation against atmospheric heat and cold, and also act as waterproof material. Waxes in the exoskeleton of insects, and cutin. An additional protective layer on the cuticle of epidermis of some plant organs, like leaves, fruits, seeds, are some of the main examples. Lipids have been classified as acylglycerols, waxes, phospholipids, sphingolipids, glycolipids, terpenoids, including carotenoids and steroids. Acylglycerols. Acylglycerols are composed of glycerol and fatty acids. The most widely spread acylglycerol is triacylglycerol, also called triglycerides or neutral lipids. Chemically, acylglycerols can be defined as esters of fatty acids and alcohols. An ester is a compound produced as a result of chemical reaction of an alcohol with an acid, and a water molecule is released. Hydroxyl is released from alcohol and hydrogen from an acid. Hydrogen and hydroxyl combine and form a water molecule. Fatty acids are one of the most important components of triglycerides. Fatty acids contain even number 2 to 30 of carbon atoms in straight chain, attached with a hydrogen, and having an acidic or carboxylic group. They may contain no double bond, saturated fatty acids, or up to 6 double bonds, unsaturated fatty acids. In animals, fatty acids are straight chains, while in plants these may be branched or rings. Solubility of fatty acids in organic solvents increase with increasing the number of carbon atoms in the chain. Palmitic acid C16 is much more soluble in organic solvents than butyric acid C4. The melting point of palmitic acid is 63.1 degrees C, as against -8 degrees C for butyric acid. Fats containing unsaturated fatty acids are usually liquid at room temperature and are said to be oils. Fats containing saturated fatty acids are solids. Animal fats are solid at room temperature, whereas most of the plant fats are liquids. Fats and oils are lighter in water and have a specific gravity of about 0.8. They are not crystalline, but some can be crystallized under specific conditions. Waxes. Waxes are widespread as protective coating on fruits and leaves. Some insects also secrete wax. Chemically, waxes are mixtures of long-chain alkanes with odd numbers of carbon atoms ranging from C25 to C35, and alcohols, ketones, and esters of long-chain fatty acids. Waxes protect plants from water loss and abrasive damage. They also provide water barrier for insects, birds, and animals, such as sheep. Phospholipids. Phospholipids are derivatives of phosphatidic acid, which are composed of glycerol, fatty acids, and phosphoric acid. Nitrogenous bases such as choline, ethanolamine, and serine are important components of phospholipids. They are widespread in bacteria, animals, and plant cells, and are frequently associated with membranes. Phosphatidylcholine is one of the common phospholipids. Terpenoids. Terpenoids are a very large and important group of compounds, which are made up of simple repeating units, isoprenoid units. This unit, by condensation in different ways, give rise to compounds such as rubber, carotenoids, steroids, terpenes, etc. Lipids constitute major source of energy and play an important role in the structure of membranes of cell and of organelles found in cell. They also provide insulation, mechanical protection, and protection from water loss and abrasive damage. Proteins. Proteins are the most abundant organic compounds to be found in cells and comprise over 50% of their total dry weight. They are present in all types of cells and in all parts of the cell. Proteins perform many functions. They build many structures of the cell. All enzymes are proteins, and in this way they control the whole metabolism of the cell. As hormones, proteins regulate metabolic processes. Some proteins work as carriers and transport specific substances such as oxygen, lipid, metal ions, etc. Example is hemoglobin. Some proteins called antibodies defend the body against pathogens. Blood clotting proteins prevent the loss of blood from the body after an injury. Movement of organs and organisms and movement of chromosomes during anaphase of the cell division are caused by proteins. Proteins are the polymers of amino acids, the compounds containing carbon, nitrogen, oxygen, and hydrogen. The number of amino acids varies from a few to 3,000 or even more in different proteins. Amino acids. About 170 types of amino acids have been found to occur in cells and tissues. Most of the proteins are, however, made of 20 types of amino acids. All the amino acids have an amino group and a carboxylic group attached to the same carbon atom known as alpha carbon, or maybe hydrogen atom as in glycine, or methyl as in alanine, or any other functional group. So amino acids mainly differ due to the type or nature of the R group. Amino acids are linked together to form polypeptides proteins. The amino group of an amino acid may react with the carboxylic group of another, releasing a molecule of water. For example, glycine and alanine may combine together. The linkage between hydroxyl of carboxylic group of one amino acid and hydrogen of amino group of another amino acid release water and a carbon-nitrogen link to form a bond called peptide bond. The resultant compound glycylalanine have two amino acid subunits and is a dipeptide. A dipeptide has an amino group at one end and carboxyl group at other end of the molecule. So both reactive parts are again available for further peptide bonds to produce tripeptides, tetrapeptides, and pentapeptides, leading to polypeptide chain. Structure of proteins. Each protein has specific properties which are determined by the number and the specific sequence of amino acids in a molecule, and upon the shape which the molecule assumes as the chain folds into its final compact form. There are 4 levels of protein organization. Primary structure. The primary structure comprises the number and sequence of amino acids in a protein molecule. F. Sanger was the first scientist who determined the sequence of amino acids in a protein molecule. After 10 years of careful work, he concluded that insulin is composed of 51 amino acids in 2 chains. One of the chains had 21 amino acids and the other had 30 amino acids, and they were held together by disulfide bridges. Hemoglobin is composed of 4 chains, 2 alpha and 2 beta chains. Each alpha chain has 141 amino acids, while each beta chain has 146 amino acids. The size of a protein molecule is determined by the type of amino acids and the number of amino acids comprising the particular protein molecule. Now we know that there are over 10,000 proteins in the human body, which are composed of unique and specific arrangements of 20 types of amino acids. The sequence is determined by the order of nucleotides in the DNA. The arrangement of amino acids in a protein molecule is highly specific for its proper functioning. If any amino acid is not in its normal place, the protein fails to carry out its normal functions. The best example is sickle cell hemoglobin of human beings. In this case, only one amino acid in each beta chain out of 574 amino acids do not occupy the normal place in the protein. In fact, this particular amino acid is replaced by some other amino acid, and the hemoglobin fails to carry any or sufficient oxygen, hence leading to death of the patient. Secondary structure. The polypeptide chains in a protein molecule usually do not lie flat. They usually coil into a helix or into some other regular configuration. One of the common secondary structures is alpha helix. It involves a spiral formation of the basic polypeptide chain. The alpha helix is very uniform geometric structure with 3.6 amino acids in each turn of helix. The helical structure is capped by the formation of hydrogen bonds among amino acid molecules in successive turns of the spiral. Beta-plated sheet is formed by folding back of polypeptide. Tertiary structure. Usually, a polypeptide chain bends and folds upon itself, forming a globular shape. This is the protein's tertiary conformation. It is maintained by 3 types of bonds, namely ionic, hydrogen, and disulfide. For example, in aqueous environment, the most stable tertiary conformation is that in which hydrophobic amino acids are buried inside, while the hydrophilic amino acids are on the surface of a molecule. Quaternary structure. In many highly complex proteins, polypeptide tertiary chains are aggregated and held together by hydrophobic interactions, hydrogen, and ionic bonds. This specific arrangement is quaternary structure. Hemoglobin, the oxygen-carrying protein of red blood cells, exhibits such a structure. Classification of proteins. Because of the complexity of structure and diversity in their function, it's very difficult to classify proteins into a single well-defined fashion. However, according to their structure, proteins are classified as follows. Fibrous proteins. They consist of molecules having one or more polypeptide chains in the form of fibrils. Secondary structure is most common in them. They are insoluble in aqueous media. They are non-crystalline and are elastic in nature. They perform structural roles in cells and organisms. Examples are silk fiber from silkworm and spiderweb, myosin in muscle cells, fibrin of blood clot, and keratin of nails and hair. Globular proteins. These are spherical or ellipsoidal due to multiple foldings of polypeptide chains. Tertiary structure is most common in them. They are soluble in aqueous media such as salt solution, solution of acids or bases, or aqueous alcohol. They can be crystallized. They disorganize with the changes in physical and physiological environment. Examples are enzymes, antibodies, hormones, and hemoglobin. Nucleic acids. Nucleic acids were first isolated in 1869 by Friedrich Becher from the nuclei of pus cells. Due to their isolation from nuclei and their acidic nature, they were named nucleic acids. Nucleic acids are of two types: deoxyribonucleic acid or DNA and ribonucleic acid or RNA. DNA occurs in chromosomes, in the nuclei of the cells, and in much lesser amounts in mitochondria and chloroplasts. RNA is present in nucleolus, in ribosomes, in the cytosol, and in smaller amounts in other parts of the cell. Nucleic acids are complex substances. They are polymers of the units called nucleotides. DNA is made of deoxyribonucleotides, while RNA is composed of ribonucleotides. Each nucleotide is made of 3 subunits: a 5-carbon monosaccharide, a pentose sugar, a nitrogen-containing base, a phosphoric acid. Pentose sugar in ribonucleotide is ribose, while in deoxyribonucleotide it's deoxyribose. Nitrogenous bases are of two types: single-ringed pyrimidines and double-ringed purines. Pyrimidines are cytosine, thymine, and uracil. Purines are adenine and guanine. Phosphoric acid has the ability to develop ester linkage with hydroxyl group of pentose sugar. In a typical nucleotide, the nitrogenous base is attached to position 1 of pentose sugar, while phosphoric acid is attached to carbon at position 5 of pentose sugar. The compound formed by combination of a base and a pentose sugar is called nucleoside. A nucleoside and phosphoric acid combine to form nucleotide. Each nucleotide of RNA contains ribose sugar, whereas sugar in each nucleotide of DNA is deoxyribose. ATP is also an important nucleotide, used as energy currency by the cell. DNA or deoxyribonucleic acid. DNA is hereditary material. It controls the properties and potential activities of the cell. It is made up of 4 kinds of nucleotides, namely D-adenosine monophosphate, D-guanosine monophosphate, D-cytidine monophosphate, and D-thymidine monophosphate. These nucleotides are united with one another through phosphodiester linkages in a specific sequence to form long chains known as polynucleotide chains. 2 nucleotides join together to form dinucleotide, whereas 3 join together to form trinucleotide. Nicotinamide adenine dinucleotide, abbreviated as NAD, is an example of a dinucleotide. It's an important coenzyme in several oxidation reduction reactions in the cell. In 1951, Erwin Chargaff provided data about the ratio of different bases present in this molecule. This data suggested that adenine and thymine are equal in ratio, and so are guanine and cytosine. Maurice Wilkins and Rosalind Franklin used the technique of X-ray diffraction to determine the structure of DNA. At the same time, James D. Watson and Francis Crick built the scale model of DNA. All the data thus obtained strongly suggested that DNA is made of two polynucleotide chains, or strands. The two strands are coiled round each other in the form of double helix. Coiling of two strands is opposite. They are coiled antiparallel to each other. The two chains are held together by weak bonds or hydrogen bonds. Adenine is always opposite to thymine, and guanine and cytosine are opposite to each other. There are two hydrogen bonds between adenine and thymine pair, and three hydrogen bonds between guanine and cytosine pair. The two strands are bound around each other so that there are 10 base pairs in each turn of about 34 angstrom units. The amount of DNA is fixed for a particular species, as it depends upon the number of chromosomes. The amount of DNA in germ cells, that is, sperms or ova, is 1/2 to that of somatic cells. All the information for structure and functioning of the cell is stored in DNA. For example, in chromosomes of bacterium E. coli, each of the paired strands of DNA contains about 5 million bases arranged in a particular linear order. The information in those bases is divided into units of several hundred bases each. Each unit is a gene, a unit of biological inheritance. The E. coli genome consists of more than 4 million base pairs, which code for at least 4,288 proteins. Haemophilus influenzae is the first microbe to have genome completely sequenced, and this was published on July 28, 1995. RNA or ribonucleic acid. Like DNA, RNA is a polymer of ribonucleotides. The RNA molecule occurs as a single strand, which may be folded back to give double helical characteristics. The nitrogenous bases form the usual complementary pairs, like cytosine with the guanine and uracil with adenine. RNA is synthesized by DNA in a process known as transcription. Types of RNA. Three main types of RNAs: messenger RNA, transfer RNA, and ribosomal RNA are recognized. All these three types of RNAs are synthesized from DNA in the nucleus and then are moved out in the cytoplasm to perform their specific functions. Messenger RNA. As the name indicates, it takes the genetic material from the nucleus to the ribosomes in the cytoplasm to form particular proteins. Messenger RNA carries the genetic information from DNA to ribosomes, where amino acids are arranged according to the information in messenger RNA to form specific protein molecule. This type of RNA consists of a single strand of variable length. Its length depends upon the size of the gene, as well as the protein for which it is taking the message. For example, for a protein molecule of 1000 amino acids, messenger RNA will have a length of 3000 nucleotides. Messenger RNA is about 3 to 4% of the total RNA in the cell. Transfer RNA. It comprises about 10 to 20% of cellular RNA. Transfer RNA molecules are small, each with a chain length of 75 to 90 nucleotides. It transfers amino acid molecules to the site where peptide chains are being synthesized. There is one specific transfer RNA for each amino acid. So the cell will have at least 20 kinds of transfer RNA molecules. Transfer RNA picks up amino acids and transfers them to ribosomes, where they are linked to each other to form proteins. Ribosomal RNA. It is the major portion of RNA in the cell and may be up to 80% of the total RNA. It's strongly associated with the ribosomal proteins, where 40 to 50% of it is present. It acts as the machinery for the synthesis of proteins. On the surface of ribosome, the messenger RNA and transfer RNA molecules interact to translate the information from genes into specific protein. Conjugated molecules. Two different molecules belonging to different categories usually combine together to form conjugated molecules. Carbohydrates may combine with proteins to form glycoprotein or with lipids to form glycolipids. Most of the cellular secretions are glycoprotein in nature. Both glycoproteins and glycolipids are integral structural components of plasma membrane. Lipoprotein formed by combination of lipids and proteins are basic structural framework of all types of membranes and cells. Nucleic acids have special affinity for basic proteins. They are combined together to form nucleoproteins. The nucleohistones are present in chromosomes. These conjugated proteins are not only for structural, but also are of functional significance. They play an important role in regulation of gene expression.